Hello everyone, we are the Waterloo iGEM and we are so excited to present our project for this year, REMINE, Closing the Loop on Heavy Metal Waste. The problem-space Waterloo iGEM chose to tackle concerns the electronics manufacturing industry. Electronics manufacturing consists of many processes, each of which generates wastewater with a unique composition of heavy metals. Contaminated wastewater is often treated by chemical precipitation, which forms a toxic sludge that is disposed of in landfills. This poses major risks to human health and the environment, and also prevents the metal from being recovered and reused. We have designed an end-of-process manufacturing waste treatment system aimed at removing and recovering heavy metal from wastewater. With our proposed solution, the released wastewater will be free of metal and decrease risks to the health of environment, wildlife, and humans. Furthermore, recovered metal ions will be purified for reuse in various industries, a feature that is not achieved through most current treatment methods.
We have developed a technology that not only removes dissolved metals from industrial wastewaters, but also recovers them for reuse in the manufacturing industry. REMINE is a packed column bioreactor that contains metal-binding fusion proteins immobilized on a cellulose matrix. As metal wastewater flows through the reactor, the dissolved metal binds to the immobilized fusion proteins, leaving clean water that can be discharged to the environment. Once our packed column reactor is saturated with metal, we can release the trapped metal ions by changing the chemical environment of the reactor using an eluent solution. This leaves the captured metal ions concentrated in a small volume of pure solution. The recovered metal can then be reused for manufacturing other products. This reduces the need to mine and purify new material, which is both economically and environmentally advantageous. Our technology is also highly modular and could be applied to a huge range of situations. However, in order to concretely demonstrate the potential of our technology, we have chosen to propose a solution to one particular waste metal situation. We will consider dissolved copper waste from a typical semiconductor manufacturing facility. This facility produces 1.48 million tons of dissolved copper waste each year, so we need to scale our process accordingly. The facility's copper waste also contains, on average, 30 milligrams per liter of dissolved copper. The US EPA regulations for copper waste say that we can release no more than 1.3 milligrams of copper to the environment, and this means that our technology will need to be able to reduce the copper concentration from 30 to 1.3 milligrams per liter.
Through this presentation, we'll walk you through the 3 different project goals of REMINE. First, to design an improved metal binding protein. Second, to design a fusion protein system. And third, to design a process for large-scale use.
The first goal of our project was to design an improved copper binding protein for our bioreactor. First, we needed to choose a suitable copper binding protein to use in our reactor. Many copper binding proteins exist, but we settled on the protein MST-CoPC. MST-CoPC is a good choice because it is small, monomeric, fully soluble, and has a single copper-2 binding site. Its sequence and crystal structure are also known, making it easy to work with. We then set out to further improve MST-CoPC's copper binding affinity. This will allow our packed column reactor to be more efficient at removing dissolved copper. Our protein improvement process has several steps, including the following. The first step is design, where we identify possible mutations that could improve copper binding. Next is our build step, where we use the software Rosetta to implement our mutations and quickly check their impact on copper affinity. And lastly is our test step, where we evaluate the copper affinity of our engineered proteins using molecular dynamics. For our design phase, we wanted to choose mutations that would improve MST-CoPC's copper affinity. We did this by reading existing literature and by examining MST-CoPC's structure using the viewing software PyMol. Some of our selected mutations are inside the copper binding site and change the amino acids that coordinate the copper ion. Other mutations are located outside the active site and have more indirect effects on copper binding, such as changing the protein's conformation and the size of the active site. 2 examples of mutations are shown here. On the left, we have the original MST-CoPC protein, and then in the middle, we substitute histidine number 85 for aspartate, changing the properties of the active site. In the rightmost image, we substitute aspartate 83 for histidine. Once we had decided on potentially useful mutations, we needed to implement and test them. We made the mutations to our protein using the protein modeling software Rosetta. Rosetta also allows us to compute an energy score for each mutant, so we can determine which of our possible mutations are the most promising. Here we can see the same example proteins as the last slide, this time with energy scores. More negative scores indicate greater copper affinity, while more positive scores indicate lower copper affinity. From these results, we can see that H85D has a better energy score than the original MST COP-C, so we will keep it for further analysis. Conversely, D83H has a worse energy score, so we will reject this mutation. From here, we can move forward with the most promising of our mutations and also try making and testing proteins that have multiple mutations in each one.
The final step in our protein engineering strategy was to apply molecular dynamics in order to test the protein candidates that we've come up with. Molecular dynamics software uses an advanced integration scheme in order to simulate the entire protein copper solvent system at an atomic resolution. An example of MST COP-C mutation H85D interacting with copper is shown on the right, where we can see the copper ion in bronze interacting with the active site. In order to compute the binding affinity of our protein copper system, we simulated each of our MST COP-C mutants interacting with copper for 10 nanoseconds using the software GROMACS. From GROMACS, we can compute the interaction energy of each protein candidate with the copper ion. The ratio of these energies determines a measure of binding affinity. Here we have some example interaction energies. As you can see, the H85D mutant has over twice the interaction energy of the original MST COP-C. In fact, a quadruple mutant we created has an even higher energy score that is 3 times the original MST COP-C. This was our best copper binding protein candidate, so we created a biobrick for it. Next steps, these proteins need to be tested in the lab using a copper affinity assay so that we can confirm that these proteins are indeed improvements on the original.
The second goal with REMINE was to design a fusion protein system wherein the metal binding protein is fused to a cellulose binding module for immobilization. When deciding which protein components to use as a cellulose binding module and what to use as a metal binding protein, we developed the following criteria to use when searching through literature, the iGEM registry, and gene databases. This led us to choose CBM2A, which was already found on the parts registry, and as Matt already mentioned, MST COP-C. Using these two components, we followed these steps to design the fusion protein and expression system. First, we removed the signal peptide sequence from COP-C, added a flexible linker between the CBM and metal binding protein, and a hexahistidine tag for purification. Lastly, we added a TEV cleavage site to remove the his tag following purification. Next, the fusion protein was modeled using iTASR, UCSF Chimera, and Modeler. This was done as a rough assessment of accessibility of the metal binding and cellulose binding sites in the fusion protein before proceeding. Lastly, an expression system was determined for the production of the fusion protein. Our team developed a cloning plan shared on our wiki for use in the PET11A vector under an IPTG inducible promoter. A chassis for expression, E. coli BL21DE3, was chosen since recombinant CBM fusions have already been expressed successfully in E. coli, and because the strain is well known in protein production. Despite being unable to enter the lab due to COVID-19, we continued to research methods that could eventually be used for the measurement and validation of the fusion. First, a copper binding method was found and adapted. This method allows quantitative measurement of metal binding affinity using spectroscopy and other basic lab tools. It can also be readily adapted to measuring the affinity of other metal binding proteins. Secondly, cellulose binding affinity will also be measured with spectroscopy-based assays, looking at the protein content in the supernatant after binding, and doing so with various incubation times. Throughout this work, designing the fusion protein, we achieved 3 biobrick goals. First, we contributed to the part documentation for CBM2A on the registry. by sharing important structural and biochemical information about this domain that we found when combing through literature. Next, we also engineered several parts, including MST-COP-C and MST-COP-C CBM2A fusion and the improved MST-COP-C CBM2A fusion, using the work from math. Lastly, we worked to improve a part by adding the flexible linker used to the CBM2A part. This will hopefully enable future teams to build more effective fusions with CBM2A.
Our last goal was to develop an industrial process that would allow our technology to be applied at a large scale. For scale-up, we have chosen to use a packed column reactor, as shown on the right. Our reactor will have a large chamber filled with cellulose, onto which our fusion protein will be immobilized. In the first stage of our reactor's operation, the copper-containing waste will flow through the reactor, and the copper ions in the waste will be trapped by the immobilized fusion proteins, causing clean water to flow out. Once the reactor has been saturated with copper ions, we proceed to the second stage of operation. In this stage, we will release the bound copper ions using a small volume of eluent solution. This will create a concentrated and pure copper solution that can be recycled for other manufacturing applications. This also resets the reactor to its original stage, allowing it to be reused many times. In order to determine if this reactor will be effective, we did some modeling using partial differential equations. The first equation describes the motion and binding of copper ions in the reactor. The left side of the equation describes transport effects, the second term representing diffusion of copper from high to low concentration regions, and the third term representing the transport of copper by fluid flowing through the reactor at a velocity v. The right-hand side of the equation determines how much copper per second binds to the fusion proteins at a given point in the reactor. This is a standard kinetic model based off of the Langmuir isotherm equation. The second equation is called the Ergun equation and describes the change in pressure across the reactor as a function of the fluid speed, the length of the reactor, as well as the porosity and density of the fluid. Other parameters which appear in this model are described in detail on the wiki. Using these equations, the model tells us how big we should scale our reactor to treat the semiconductor manufacturing waste stream we discussed earlier. After applying a quasi-steady state approximation to the transport model, we simulated the rate at which copper ions are absorbed onto the reactor. As a result, we found that over 99.5% of copper ions are bound within 5 seconds of entering the reactor. This allowed us to calculate that the reactors should be 1.28 meters in diameter and 12.8 meters in length, with a flow rate of 46.9 liters per second. To achieve this flow rate, we should pressurize the reactor inflow to 0.126 bar, which is a very reasonable pressure. Our model shows that a reactor with these parameters can reduce the copper concentration from 30 milligrams per liter to under 0.1 milligrams per liter, which is well below the US EPA requirement of 1.3 milligrams per liter.
Once we had designed our reactor, we then wanted to think about how we would implement it in an industrial setting. Our example copper-containing semiconductor waste stream contains a variety of components in addition to copper. We could remove these other components using various pre-treatment steps based on existing technologies. This would leave us with just the copper to remove using our packed column reactor. Before the packed column reactor, though, we would want to adjust our waste to pH 7.2, since this is the optimal pH for copper ions to bind with our fusion proteins. We can treat just under 850,000 liters of waste over a period of 5 hours before the reactor becomes saturated with copper. After that, we'll move on to stage 2, where we will elute the copper from the reactor. We've shown that this can be accomplished using a pH 4.8 eluent solution that will cause the copper to unbind from the fusion proteins, leaving a concentrated and pure copper solution that can be recycled. We expect the solution step could be accomplished in less than 1 hour. This timeline allows us to achieve 4 reactor cycles a day, which will allow us to achieve our goal of treating 1.48 million tons of semiconductor manufacturing waste each year.
We developed REMINE in such a way that our system would not pose any biosafety risk. Since our final packed column does not contain any living organisms, biocontainment strategies do not need to be implemented. The other materials in our column are also not a biological or environmental hazard, because the metal binding proteins would be considered non-hazardous and safe to dispose of normally by typical laboratory standards. And cellulose, being a natural derivative of wood, decomposes easily in the environment. Most types of similar chemical biological waste are not classified as hazardous by regulatory bodies such as the EPA. Therefore, no specialized disposal protocols need to be implemented, and the use of our system should not pose any significant biological chemical hazard.
Regarding human practices, we collaborate with Untitled iGEM on the protein design and molecular dynamics aspects of our project. We also collaborate with Ohio State iGEM on the modeling aspect of their project. In July, we hosted a Canadian-wide virtual social hour to encourage communication between all members of Canadian teams, meet new people, and build our local synthetic biology community. In terms of education, Waterloo iGEM was able to interact with high school students through an engineering outreach session hosted by our institution called Catalyst. We also developed various workshops for our and other teams on the foundational concepts in synthetic biology and created resources pertaining to bioinorganic chemistry and an introduction to transport phenomena.
For excellence in another area, Waterloo iGEM spearheaded two main projects. First, we worked on developing an open source accessibility toolbar for iGEM wikis that currently has a read aloud function and font size functions. For the future, we aim to include a word highlighter to complement the read aloud function and a customizable color palette for different types of color blindness. Our second project involved researching barriers to inclusion, equity, and diversity in STEM. Our research looked at two things: how academic streaming in Ontario prevents racialized folks, primarily Black individuals, from entering STEM fields as a whole, and we also observed the importance of representation. As iGEM teams, we have the ability to learn about the systems of oppression impacting fellow iGEM members, folks outside of iGEM, and individuals everywhere, and we should hold ourselves accountable to tailor our outreach and education to bridge the gap between advocacy and academia.
The next steps for REMINE include 3 parts. First, we will validate our improved proteins in the lab by experimentally measuring their binding affinity. Second, we will construct a bench scale prototype of our packed column to test its ability to remove metal ions. Third, we will make our system more modular and customizable by designing proteins to target other metals, such as those found in specific industrial wastewaters or those that are rarer and more valuable. In summary, we successfully built a proof of concept for removing copper waste from an example semiconductor facility by modeling a REMINE system and tripling the binding affinity of a copper binding protein. We also created and shared educational materials that cover many different perspectives, whether that was teaching students about how the engineering process is applied in synthetic biology, or educating iGEM teams how they can be more inclusive in their recruitment, project goals, and wiki. We hope that we reminded you of the importance of closing the loop for heavy metal waste, along with communicating our research to a wider audience. Thanks for listening.